Alright, this is a video lesson for Unit 13, Meiosis, as we go through and talk about how gametes are formed, okay? So make sure you are writing these notes down on your guided notes, as well as adding anything extra that I write down, and listening to what I say. Make sure you rewind, rewatch any parts that you are confused on. Alright, so let's get started. Right, the process of meiosis is a form of cell division by which gametes with half the number of chromosomes are produced. Okay, so a gamete, a little bit of a vocab lesson here first. Gamete, this is simply a sex cell or reproductive cell, okay? And so this could be either a sperm cell if you are a male or an egg cell if you're a female, okay? So that's all a gamete is, sperm or egg. Right, these sperm or egg cells, or gametes, are going to have half the number of chromosomes. Okay, so half the number of chromosomes so that when they combine, we're going to get a full set. Right, so two more vocab words I want to go over before we go into any more notes are diploid and haploid. So a diploid cell, right, is a cell with a full set of chromosomes, right? In humans, this is 46, okay? So in humans, the magic number of chromosomes is 46 in a human cell, right? This is only going to be in body cells, or our fancy word for that is a somatic cell. So a body cell would be like your skin cell, muscle cell, stomach cell. All of those are body cells. All of those have 46 chromosomes. Okay, that's a full set. Now, a haploid cell, on the other hand, has half the number of chromosomes. So what is half of 46? Well, it's 23. So these cells are going to have 23 chromosomes. Okay, so our haploid cells are going to be our gametes, or our sperm and egg cells. Okay, they're going to have half the number of chromosomes, and we'll find out why that's important in just a little bit. Now, meiosis is crucial for sexual reproduction to occur. We have to be able to produce gametes so that we can combine them and get a living, viable offspring. And to get these gametes, we're going to go through two different sets of divisions, meiosis 1 and meiosis 2. Right, so at the bottom you see a picture. You can see here that we have a sperm cell that's haploid with 23 chromosomes, an egg cell that's haploid with 23 chromosomes. When we combine these two, we get a zygote, or a fertilized egg, that has a total of 46 chromosomes, so that making a diploid cell. So we put two haploid cells together to create a diploid cell. This is the process of fertilization. This is the ultimate goal, right, after meiosis. All right, so meiosis, we are producing gametes, or sperm or egg cells. And these gametes, like we've established, are going to have half the number of chromosomes as a regular body cell. Right? This process only occurs in the gonads. So if you're a male, this is going to occur in the testes. If you're a female, this process occurs in the ovaries. In males, we call this process spermatogenesis, okay, making some sperm. And in females, we call this oogenesis, which creates the egg or the ova. Right, so let's break down spermatogenesis. So in spermatogenesis, we have a human sex cell, right? So we start out with a diploid cell with 46 chromosomes. Through the process of meiosis 1 and meiosis 2, we result in 4 different sperm cells that all are haploid and have 23 chromosomes. Okay, so in spermatogenesis, we are getting 4 sperm cells that are created. In oogenesis, we start out again with a diploid cell containing 46 chromosomes. This, we go through meiosis 1 and meiosis 2, but at the end, we only result in one egg that is haploid. The other 3 are turned into what we call polar bodies that are going to end up just dying. So they don't actually mature into or turn into an egg. Okay, so in oogenesis in females, we produce 1 egg through the process of meiosis. And actually, this process all happens before the female is even born. So girls are actually born with all of the eggs that they are ever going to have. Okay, remember all cells go through a process of interphase, and this is just when the cell is growing normally and having its normal process. So the interphase of meiosis is very similar to the interphase of mitosis. Our chromosomes replicate in the S phase just like before, right? But now we're going to have each chromatids. Remember, we still have those sister chromatids that are identical, attached at the centromeres just like before. We also are going to be replicating our centrioles. So this is when we go from a single-stranded chromosome to our double-stranded chromosome that's going to look like the X now, and you can easily see the centromere in the middle where those sister chromatids are connected. Okay, so here is like a cell in interphase. Remember that the chromosomes are not visible yet. We can easily see our nucleus and our nucleolus inside of our cell. So this is something you should already know and be familiar with. Right, meiosis 1 is going to occur in 4 phases, and this is when we're going to reduce our chromosome number by 1/2. Okay, so this is important. I want you to highlight this, or box it, or star it, or something. So after meiosis 1, okay, this is when we go from diploid to haploid cells. At the end of meiosis 1, we're going to have haploid cells, no longer diploid. Meiosis 1 is going to have 4 phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. So these sound very similar to those phases that we have in mitosis. Okay, in prophase 1, this is our longest and most complex phase. We have a lot going on in this phase. This is when we're going to see our chromosomes condense, and we're going to see synapses occur between homologous chromosomes to form what we call a tetrad. So the prefix tetra means 4. All right, we're going to talk about what a homologous chromosome is coming up soon, so let's just keep that vocabulary in the back of our mind. All right, so synapses is when these come together to form 4. So a tetrad is 2 chromosomes or 4 chromatids. So this is going to be 2 sister chromatids and 2 non-sister chromatids. So let's take a look on the next slide at what I mean. Right, so you can see here there is a tetrad, and we'll have a better illustration of what's happening here coming up. Right, so non-sister chromatids are called homologs. So just like you have 2 parents, a mother and a father, each parent is going to contribute 1 of the homologous chromosomes, okay? These homologous chromosomes contain DNA instructions for the same genes. Now, these genes are not going to be the exact same, okay? So, for instance, if we're talking about eye color, right, mom may contribute blue eye color, dad may contribute brown eye color. Now, it's still instructions for eye color, right? But it's just a different form of that gene. So that's what we mean when we talk about homologous chromosomes. So in synapsis, we have these 2 homologous chromosomes. So we have a maroonish looking one and a yellow one. And these both are made up of sister chromatids. So our maroon one is 2 sister chromatids, and our yellow one is 2 sister chromatids. When we put these together in prophase 1 in synapsis, this forms a tetrad, and you can count them here. You can easily see there is 1, 2, 3, 4 chromatids, right, making up this tetrad. These 4 chromatids consist of 2 homologous chromosomes. Okay, so again, a homologous chromosome is a pair of chromosomes, maternal and paternal, right, from your mother and father, that are similar in shape and in size. So they carry genes controlling the same traits. Each position of a gene is in the same place on each of the chromosomes. So you're going to have 23 pairs of homologous chromosomes, 23 total pairs. The first 22 are called autosomes. And these chromosomes here, autosomes, these are just like your regular body traits. Eye color, skin color, height, freckles, no freckles, brown hair, blonde hair, etc. Okay, the last pair, our 23rd pair, are our sex chromosomes. And this is going to tell whether you are male or whether you are female. Right? So here is an illustration of homologous chromosomes. So if you look closely here, we have one homologous chromosome, the maroon one. Let's just say this is the mother, and the yellow is from the father. On each of these homologous chromosomes, they each contain the gene for eye color, right? Notice that this is at the same spot on both of the homologous chromosomes. In addition, if you look here, the bottom, they also have the gene for hair color. Again, this is in the same position on both of the chromosomes, okay? These are not going to be identical. They're going to be slightly different, but they still code for the same basic traits. Right. Something else important that happens during prophase 1 is something called crossing over. And this happens between the non-sister chromatids of our homologous chromosomes. This is going to be whenever segments of our non-sister chromatids break and reattach to the other chromatid. This will be easier to see whenever I show you a picture of it in just a second. This is going to cause more genetic recombination, right? This is a super important concept because crossing over introduces new combinations of genes that didn't exist before, right? And from an evolutionary standpoint, that's a pretty big deal. Okay, so here is our illustration. We have our two homologous chromosomes. Notice here I have labeled for you the non-sister chromatids. So a non-sister chromatid means it's a chromatid on each of the homologous chromosomes. So they are not the same. We have a maroon one and we have a yellow one. They're going to cross over. When crossing over occurs, you can see they're basically just swapping a piece of the chromatid, non-sister chromatid, with the other one. So the result is here. We now have part of the yellow on the maroon one, and now the maroon part is on the yellow one. Again, this has just recombined the genes into a different order, right, which is going to add more genetic variation. Right, here's another picture of this process. You can see here crossing over happening between these two homologous chromosomes, our paternal and maternal. Crossing over happens, and the result is now we have swapped pieces of the chromatids, so now each chromosome has a different combination than what it started with. Right, sex chromosomes. If you are a female versus a male, females are XX, okay? X's are bigger than Y's because the Y chromosome doesn't contain that much information. It just is going to tell whether or not the organism is male or female. So if it is a male, it's going to have XY, and the Y chromosome is just a smaller version of the X. Right, in meiosis 1, prophase 1, the nucleus and nucleolus disappear, our spindle forms, right, and crossing over happens. So the main thing I want you to know is that we have synapsis, we see our tetrads, and we cross over during prophase 1. Right? Those are the important things that you need to know from this. In metaphase. In metaphase 1, this is our shortest phase, and this is when we are lining up those tetrads along the equator. Okay? This is also when independent assortment occurs. So you need to highlight this, star it, or something. Okay, this is important. Independent assortment occurs, right? This is means that these homologous chromosomes, they line up in a random order. So we could have like big A, big B on this side, and little b across from it, or it could have said, we could have had big A, little a, and then next to it, instead of having a big B, we have a little b, and then the big B on the other side. So these line up randomly, okay? There's no set order on how the chromosomes line up down the middle of the cell. It happens randomly. That's independent assortment. This causes more genetic recombination and genetic variation. Okay, so again, here's another illustration of the options. They can line up either way, and it's just completely random. That's independent assortment. To find out how many combinations, we use the formula 2 to the power of n. I probably won't have you do this, but in case you had to do that, then we would know that n is the number of total, n is the total of chromosomes, so 2 to the power of 2 would be 4 combinations. If we wanted to know how many combinations of sperm a human male could produce, we would simply say 2 to the power of n. We know that n is going to be 23, so 2 to the 23rd is approximately 8 million different combinations that can be created, simply due to independent assortment. Right, anaphase 1 is going to be where our homologous chromosomes separate. Okay, homologous chromosomes separate. You need to highlight this or star it, because this is important. Homologous chromosomes are separating in anaphase 1. Right? Now, keep in mind that these are just homologous chromosomes. Our sister chromatids are still attached to centromeres. So the result is going to look something like this. We have our sister chromatids still attached, and we have our homologous chromosomes have now separated to opposite sides, but they still look like X's. That is going to be anaphase. So at the end of telophase 1, we're going to still have the chromosomes looking like an X, and they're going to be now haploid cells, right? So it'll look something like this at the end of telophase 1. We'll have 2 haploid cells. We now have 2 chromosomes instead of 4. Okay, so meiosis 2, meiosis 2 is basically mitosis. We have no interphase 2. Right? We don't replicate DNA again. And the steps are almost identical to the steps of mitosis, right? So you should already know this part. Prophase 2 is same as prophase in mitosis. Simply, we're going to see our chromosomes, spindle forms, nucleus disappears. Metaphase 2 is the same as metaphase in mitosis. Our chromosomes line up down the middle of the cells. Right? Remember now, instead of having one cell, we're doing this with 2 cells at the same time. That's why I have 2 cells pictured on the screen. So metaphase, they line up down the middle, metaphase 2. Anaphase 2, the same as anaphase in mitosis, which means our sister chromatids now are going to separate. So I need you to highlight that right here. Sister chromatids separate. Okay, so in anaphase 1, our homologous chromosomes separated. In anaphase 2, the sister chromatids separate. So now it's going to look much like the anaphase of mitosis. Now we have single-stranded chromosomes. Telophase 2 is the same as telophase in mitosis. We start to see our nucleoli reappear, followed by cytokinesis. And this is going to produce 4 different haploid cells called gametes. So this results in, here's my illustration, 4 different cells. Now if you look here, these are 4, let me write a different pen, 4 genetically, write this down, unique haploid cells. Okay, if you look at this picture, none of the cells are the same. They all have a different genetic combination. Okay, so we are not getting identical cells. So here is the process of meiosis, a little animation for you. You can see here crossing over happens. That's the end of meiosis 1, and now we have meiosis 2. We're simply going to separate the sister chromatids now in meiosis 2. and then we have telophase and cytokinesis. So we get 4 different haploid cells. The whole point of this is so that we can have genetic recombination, genetic variation, right? This is important because this is going to be something that helps for natural selection, which I'm sure you've heard of, and we'll talk more about later in our unit for evolution. Now, this is why we are not alike, okay? Even you and your siblings have different genetic combinations because of this process of meiosis. And remember the definition, just kind of introducing this to you now, but we'll talk about it again in the future, is that natural selection means the strongest or the most fit are able to survive and reproduce and pass on their traits. And this will be important because we get new traits as we recombine the genes in a different way each time. Right, a couple of questions. What are the 3 sources of genetic recombination? I talked about 3, crossing over, independent assortment, and random fertilization. So we never know which egg is going to be fertilized by which sperm cell. It is completely random, okay? So we could get a new combination with each pregnancy and with each child. A 4th one is also going to be mutations, which we have talked about, and mutations, remember, can introduce new genetic combinations as well. Last question, a cell containing 20 chromosomes at the beginning would produce cells with how many chromosomes? So if 20 is a diploid number, remember, and that's at the beginning, at the end of meiosis, we're going to have half that amount. So we should have 10 chromosomes at the end of meiosis because we're halving that number. Right. Fertilization is when we're going to combine a sperm and an egg cell to create a zygote, which is a fertilized egg. So we have an egg with 23 chromosomes, a sperm cell with 23 chromosomes. This gives us a zygote then with 46 chromosomes, all of the genetic information needed to turn into a human baby. Okay, I want you to see if you can answer this question on your own. And make sure you have all of your notes filled in. Go back and rewatch any parts that you might need to rewatch, and make sure that I check off your notes when I ask to see it. All right, there is nothing else, then I think you should be good to go on meiosis.